Pesticide Residues Rapid Test Kit. We would like to introduce the Pesticide Residues Rapid Test Kit provided by Shenzhen Bioeasy Biotechnology Co Limited. It is applicable for detecting high levels of organophosphorus and carbamates pesticide residues in vegetables and other edible agricultural products. This detection method is based on the enzyme inhibition. Take out the test kit from refrigerator. The Pesticide Residue Rapid Test Kit consists of a manual, buffer solution, chromogenic agent, substrate, and enzyme reagent. After taking out the reagent from the refrigerator, wait until it reaches room temperature. The experiment is divided into 4 parts: buffer preparation, sample preparation, testing, and interpretation of the results. First, we need to prepare the buffer solution. Take 1 milliliter of buffer into an empty bottle, measure 100 milliliters of purified water, and pour it into the bottle as well. Mix well. The solution will be used as the buffer solution for our testing. It should be stored at room temperature. Next, sample preparation. For leafy vegetables, when sampling, the leaves should be folded, then cut from the leaf edge and cut into pieces. Weigh 2 grams of the sample, put into a centrifuge tube. If there is no centrifuge tube, sample cup can be used instead. For fruit, peel and cut into pieces. Weigh 4 grams of the sample, put into the centrifuge tube. Mark the sample. Add 10 milliliters of buffer solution, shake for 1 minute, then put in a standing position for another 1 minute. Place the folded filter paper in the funnel, move it to a new centrifuge tube, and pour the sample solution for filtration. The filtrate is the sample to be tested. Sample testing requires YR600 Plus reader. To turn it on, press the red button. Input your username and password, then log in. Open the Spectrophotometer module, select the pesticide residue project, fill in the sample name. Select the whole according to the number of samples. Click Start Detection. 0. First, perform a control test. Take an empty cuvette, add 2.5 milliliters of buffer solution, add 100 microliters enzyme reagent, and 100 microliters chromogenic agent. Mix with pipette. Keep in standing position for 10 minutes in order to complete the reaction. At the end of the time, add 100 microliters of substrate. Mix it quickly and put in the reader. Place the cuvette in the A1 detection hole and cover the lid. Wait for 5 to 6 seconds. Press Control button for the control test after the absorbance value rises steadily. Get the control results. The delta ABS value of the control must be more than 0.1. Press back to return to the interface. Take out the cuvette. Next, we carry out the sample testing. Add 2.5 milliliters of the test solution to the cuvette. Add 100 microliters of enzyme reagent and 100 microliters chromogenic agent. Mix with pipette. Keep in standing position for 10 minutes in order to complete the reaction. At the end of the time, add 100 microliters of substrate. Mix it quickly and then put in machine for test. After zeroing, place the cuvette in the detect hole and cover it. Press the sample button for the sample test after the absorbance value rises steadily. Get the sample test results. Press Print. Interpret the results. The sample test results are expressed as suppression rates. According to GB T 5009.199 provisions, when the suppression rates are less than 50%, it is judged that the pesticide residue is qualified. When the suppression rates are greater than 50%, it is judged that the pesticide residue is exceeded. We recommend to retest in case of positive samples.